professor at the University of Colorado in Denver, the Department of Biochemistry and Molecular Genetics. So Angelo's academic background includes a bachelor's, master's, and PhD from the University of Tuscia in Italy, which he then followed up with postdoctoral training at the Beatson Institute for Cancer Research in Glasgow, Scotland, the University of Tuscia, and the University of Colorado in Denver. Angelo is extremely well funded as the PI of multiple NIH R01 grants and is an exceptionally prolific researcher with 666 peer-reviewed papers. So I think I think that would qualify for the category of productive. Without further ado, he's a world-leading expert in red blood cell metabolism. I'm really happy to have him with us today. Angelo, welcome.
Thank you so much. Thanks for the invitation to present here today. Thanks for coming. I will be talking a lot about red blood cells today. Hopefully, it is a subject that will interest a lot of you working in, here at LSI and Center for Blood Research. Before getting into the details of the presentation, a few disclosure first. And multiple times during the presentation, I will be using the plural pronoun. We did this, we did that. When I say we, it's actually the people on this slide who actually do all the work that I will be presenting today. And I also like to say they also get to have all the fun with the instruments. But hopefully you will see their faces alive throughout the presentation. So working in the Department of Biochemistry and Molecular Genetics at the University of Colorado and playing around with a lot of expensive toys, I need to convince my department chair and everybody in the Cancer Center at the LSI University that is supporting the mass spec infrastructure we have in the lab that studying red blood cells is a worthwhile endeavor. And usually I start by sharing a few numbers. I don't know how many of you actually work with red blood cells in particular, but if you were to count all the cells in the human body in an adult, 175 centimeter tall, 75 kilos of weight, approximately 83–84% of the total cells in that individual, 25 trillion out of 30 total, 30 trillion total estimated cell in the human body are red blood cells. I'm a person who studies metabolism, and metabolism from a thermodynamic standpoint is using small molecule substrates to build macromolecules. And from an energetic and thermodynamic standpoint, red blood cells represent a unique challenge to the organism, because not only there's a lot of them, but they have an average lifespan of 120 days. So every 120 days, you remove from the bloodstream all 25 trillion, and on a daily basis, you synthesize de novo through erythropoiesis, approximately 0.2 trillion red blood cells. So a lot of the energy and small molecule substrates that the body uses goes towards building that biomass and recycling the biomass to build new red blood cells, including the iron in the prosthetic group of hemoglobin that contributes to the main function of red blood cells, transporting oxygen and gases through the human body. I'm also a metabolomics person, as I said, and red blood cell offer a unique simplified model to investigate metabolism and the protein metabolism interaction in response to external stressors in the absence of de novo gene expression. The absence of nuclei and organelles makes the theoretically red blood cell proteome rather stable endeavor. You all know that during the process of erythropoiesis, red blood cells to lose nuclei and organelles to maximize the amount of hemoglobin molecules per cell, which adds up to approximately 250 to 270 million molecules of hemoglobin per each erythrocyte. With one iron heme per prosthetic group of the hemoglobin tetramer, a full oxygen saturation red blood cell carry approximately up to 1 billion molecules of oxygen per cell. Every time an oxygen molecule jumps on and off hemoglobin, you have 1 in 10th, in some cases 1 in 8 chances of the oxygen stripping an electron off the iron. And as a result, you catalyze the formation of reactive oxygen species through Fenton-Haber-Weiss chemistry. In other words, not only red blood cells are a unique model to investigate energy metabolism, but also a unique model to investigate responses to oxygen stress in the face of a sustained amount of reactive oxygen species generation. And also in the face of the incapacity to replace damaged protein components with de novo gene expression. So I try to convince you that red blood cells there's a lot of them, and they're really loaded with hemoglobin, but historically they've been referred to as hapless sacs of hemoglobin. And the development of proteomics technologies, especially in the last 2 decades, have allowed us to appreciate the unprecedented complexity of the red blood cell proteome, which beyond hemoglobin, which makes up approximately 90% of the dry weight, 98% of the cytosolic proteome, harbors several thousands of proteins. We argue that if the final number hasn't been agreed upon by the community, but there are some studies over the last few years that argue, well, like you're seeing all these proteins because your mass spec are becoming so sensitive that you're now covering several thousands of proteins, and there's a lot of contaminants there. In reality, red blood cells have approximately 1,000, 1,500 proteins. But then, other groups use the latest and greatest mass spectrometers and threw in whole blood, Buffy-coat depleted whole blood, and they see, over 5,264 proteins. So where's the truth? Trying to tackle this question, in our lab, we collaborated with Dr. Crystalline Hudson at Columbia University, New York. We obtained Buffy-coat depleted, leukocyte filtered with log4 removal of white blood cells, log 2.5 removal of platelets. And then we additionally spent over almost 3 weeks on a flow sorter to obtain 5 microliters of ultra pure red blood cells by flow sorting out residual white blood cells, residual platelets, residual reticulocytes, and only have ultra pure mature red blood cells, CD35 positive, CD71 negative, thiazole orange negative, so devoid of mitochondria. There is a whole side caveat to that story. We actually do observe up to 5 to 7 residual mitochondrial mature blood cells in sickle cell disease, but that's a tale for another day. So ultimately, we obtain very pure ultra pure red blood cells of population. We can literally throw at them everything but the kitchen sink, gel CMS, IPH reverse phase fractionation, orthogonal method to enrich for the insoluble membrane fraction through chemical digestion. And ultimately, what we do observe is 3,775 unique proteins in the mature blood cells, which compared to all the studies in the literature are approximately 1,000 more new proteins that have never been reported, but also around 1,200 proteins that we identified to be potentially contaminants in previously published literature, including several papers from our group. So, of course, we are not only interested in understanding what's in the red blood cell proteome. If you want to, we actually created a database for those of you who like thrillers from the 1970s, 1980s. Deep Red by Dario Argento is a famous thriller. And we built this database that is freely accessible online, including a series of fine mapping of multiple post-translational modifications of potential biological relevance. On top of that, of course, the database contains the estimated copy number per cell and linkage to Unicode IDs for each protein. What we found to be really interesting to us is that beyond cataloging what proteins are present in the mature erythrocyte, we want also to understand how this protein interact with each other. So one way we went about that, other than performing preliminary fractionation enrichment for endogenous cross-links by transglutaminase 2, which is very abundant in red blood cells, was also to use cross-linking agents like D6 sulfoxide or DMTMM, which introduce covalent bonds within epsilon amine groups of lysine and carboxylic groups of aspartate and glutamates within, in our case, with a zero-length cross-linker up to 20 angstrom distance between each other. So in other words, we found a core interactome that orbits around the band 3, the most abundant membrane protein in membranes, approximately 1 million copies per cell, the titin cytoskeleton interactome, and the cytosolic interactome of hemoglobin, which in part is justified even just by hemoglobin bouncing off other molecules by chance alone. We're using all this information to catalog the in silico map of the red blood cell metabolome, identifying all the established enzymatic reaction, the catalytic components of all of them, and ultimately mapping how the red blood cell metabolism is regulated at the least at the protein level, and we're now using these maps to predict how red blood cell metabolism responds to perturbations such as blood storage, and using all of this to come up with novel storage additives for blood transfusion purposes. Of course, all sorts of applications, but I'm glad to chat more about specific details that I will not cover through this presentation today. In short, what we are trying to, we are proposing kind of from a provocative standpoint is that red blood cells represent a sort of circulating organ. If you were to weight it, it is approximately 2.5 kilos in weight, that at least they harbor proteins with activities that were previously thought to be unique to specific organs like liver, spleen, in some cases even neurotransmitter synthesis and receptors on the red blood cells themselves. At the very least, we believe that red blood cells do harbor the enzymes that could catalyze all sorts of activities that I'm listing here. And how do we go about testing whether these enzymes are actually there and working? One way we go about that is, for example, by performing mass spectrometry-based metabolomics. It's pretty much the bread and butter in our lab. If you were to lyse red blood cells and analyze through a mass spec or a gradient of time, you will see the mass to charge ratios here on the y-axis, which is what the mass spec measures, ions. In this case, you're looking at positive ion mode analysis of red blood cell metabolome, and it's dominated by approximately 50-60 peaks of metabolites that are very abundant in the millimolar level, hundreds of micromolar and tens of micromolar, like amino acids, NAD, all the way to glucose and lactate in the millimolar level. But also you see like these little dots down here in jargon in the grass that you can zoom in by cutting out 90% most abundant metabolites, and ultimately assign a name to this several thousand unique chemical formulae based on standard libraries we have available in the lab, several tens of thousands of metabolites. Of course, there are publicly available databases we can interrogate as well. One advantage of some of the platform we built in the lab is that we have methods as quick as 1 minute per sample for kind of a ballistic method based with a short chromatographic column approaches that allow us to perform population scale or metabolomics analysis for all sorts of application, including population studies in both humans, animal models, and other model organisms. I did not mention this, but of course we can run lipidomics as well, all sorts of lipidomics method. And even in this case, we developed a series of orthogonal ultra high throughput and high throughput method, 5 minutes per sample, 7 minutes per sample, that allow us to run approximately 1 to 1.5 million samples a year, depending on the year. If you're interested in reading more about this, there's a whole thematic book I edited for Springer Nature, Methods of Molecular Biology. There is the second edition coming out. If anybody has anything to contribute, I would welcome any additional contribution than the one out there. So since we are, I'm addressing the Center for Blood Research here at Life Science Institute, and I'm gonna almost exclusively be talking about red blood cells today. I just put together this slide just to summarize how we are using very similar combination of metabolomics and proteomics approaches and functional genetic studies and high throughput screening and primary samples from patients and so on and so forth to build maps of, for example, metabolic vulnerability in primary AML, acute myeloid leukemia samples from patients. If you're not working on red blood cells or not working on leukemia, replace that with a biofluid of interest or the cell type of interest or the tissue of interest, and we can apply very similar workflows in any and all of those matrices. In addition to being really fast, we want to democratize access to the platform. One hurdle we encountered over the years, especially with rural community or underrepresented minorities in some of the underrepresented communities in science in the United States, was access to, for example, venipuncture and phlebotomy to obtain samples in remote area. We live in Colorado, there's people in the mountains in the Rockies that don't want to come all the way to Denver, 3 hours, 5 hours by car to go and get blood drawn to perform this analysis. So can we send them a device that we can put on the arm, push a button, collect a dry blood spot, and then a dry blood spot can be shipped at room temperature for several weeks inside a specific container and not observe any deviation to, for example, established marker. And we use this, we actually deployed this workflow for not just for lactate measurement, but for full-blown metabolomics analysis for several studies at population scale, including, for example, recreational, semi-professional, all the way to professional and elite athletes, including, the team that won now 4 of the last 6 Tour de France in cycling. And hopefully I'm about to receive 36,000 samples from the last 3 Olympics from all sorts of sports through collaboration with the Fédération Internationale de Médecine du Sport. So what are we using this technology for? Well, we're using it to study red blood cell for transfusion purposes, and we'll get that in a second. But, one very attractive series of investigation in Colorado being Mile High City, 1,560 some meters. We have access to a lot of studies that focus on red blood cell responses and in general physiological responses to high altitude hypoxia. And just to give you a few examples of how we can use this platform and combining metabolomics and proteomics to learn new biology and interactomics to learn new biology. For example, in this study, we collaborated with the Altitude Research Center in Denver. They enrolled like 21 volunteer students from Oregon, and they flew them on what they thought was going to be a vacation all the way to the mountains. Little they knew that they were going to be put on oxygen mask all the way to La Paz, Bolivia, which is already very high. And they kept them on oxygen mask all the way to the top of Mount Shackletai at 5,300 meters. Once they made it to the top, what they do, took the oxygen mask off. There are literally people rolling on the ground. They're like altitude sickness all the way to extreme altitude sickness. Some people are puking. Best part of the experiment, but also the worst part is that all of them within hours acclimatized. All of them started doing much better within 1 hour to 6 hours. They started walking uphill after 1 day. They started running uphill after 7 days. All of this before we actually observed any changes in hematocrit. Like we started making more red blood cells, which usually take around 7 to 10 days. So what happens in the process? Well, red blood cells, despite lacking nuclei and organelles, can adapt to the environment. And one way they go about that is by synthesizing 2,3-bisphosphoglycerate, which promotes the stabilization of the oxygenated state of hemoglobin in this cohort and other studies that we've done more recently in another high altitude study in La Rinconada, Peru. And ultimately, this promotes oxygen offloading. This was established in the 1970s, but it wasn't really clear what were the mechanisms behind this metabolic adaptation. So how do red blood cells sense this process? And through a combination of the interactomic studies that I was talking about before, by studying how the differential protein-protein interaction chain responds to hypoxia within red blood cell membrane, we found that the interactome of the most abundant membrane protein, the BAN3, or an ion exchanger 1, changes in response to hypoxia. In particular, under the oxygenated condition, the oxyhemoglobin increases its migration it migrates to the membrane and increases its interaction with the otherwise intrinsically disordered region of the N-terminus allosolic domain of BAN3. And in this process, glycolytic enzymes such as GAPDH which are otherwise bound and inhibited by the binding to BAN3, are released from the membrane to the cytosol. And in the process, they are activated. GAPDH is right upstream to 2,3-BPG synthesis through the rapoport-Lubrin shunt, which then catalyzes or contributes to modulating the allosteric of deoxyhemoglobin and oxygen offloading. So through a combination of animal models, studies in humans, and genetic and pharmacological manipulation, we ultimately ended up publishing a series of studies in collaboration with Dr. Yang Sha and many other colleagues, formerly at the University of Texas Houston, University of Changsha in China, where we identified that red blood cells not only sense and release adenosine triphosphate in response to hypoxia, but this ATP is broken down to ADP and adenosine signal through adenosine receptor A to B, and this very process promotes its own phosphorylation, ubiquitination, removal from the membrane of the ENT1 transporter. So now you accumulate adenosine much faster the second time you go to high altitude, if you were to descend to lower altitude and reascend again. Red blood cells can synthesize de novo the transporter for adenosine. So the red blood cells that were exposed to high altitude hypoxia the first time until they are removed from the bloodstream, usually weeks to up to 120 days later. And then that means that even though you don't have a transcriptional upregulation of signaling pathways, you have a post-translational series of modification or regulatory processes that give you a memory of the acclimatization to high altitude hypoxia and allow you to acclimatize much faster in the future. I mentioned already the deoxyhemoglobin GAPDH release and promotion of the bisphosphoglycerate mutase-dependent generation DPG, but you also found a series of other pathways that involve sphingosine-1 phosphate synthesis and further contribution to deoxyhemoglobin allosteric, and transglutaminase 2-dependent ubiquitination of key lysins in BPGM that prevents transglutaminase 2-dependent formation of isopeptide bond that prevents the otherwise ubiquitination of the same lysins and untoward removal of BPGM. So there is a series of mechanisms. I will not go much into the details here today, but will suffice to say that this is relevant for blood storage, as I will show you in the second part of the presentation, but it is relevant for human biology at large. For example, in collaboration with Isha Jain and Alan Doctor, I think this was like a month and a half ago, we published in Cell Metabolism that the red cell organ is actually really good at keeping in control, in check, circulating levels of glucose. And it all started with the observation that people living at high altitude are much less likely to develop diabetes, which has been observed like almost retrospectively, but has never been investigated directly from a mechanistic standpoint. And through a series of studies involved bleeding, transfusion, stimulation of erythropoiesis, and so on and so forth, we identified that red blood cells contribute to a good chunk of the unexplained glucose oxygen consumption by fMRI and in vivo tracing experiments, and that this process was indeed regulated by the very same GAPDH, the oxygen-dependent deoxyhemoglobin BAN3 interactome that releases GAPDH from the membrane to the cytosol in hypoxia, thereby promoting bisphosphoglycerate synthesis. So this ends the first part of the presentation, and now I'm finally talking about something that you may be more interested in from a transfusion medicine standpoint, looking at the blood center colleagues here in the room. All of you, I'm pretty sure, know that red blood cell not only are the most abundant cell in the human body, but also it's the most common, commonly issued product, the most common in hospital medical intervention, say after vaccination, even though not necessarily vaccination may occur in the hospital. Approximately 100 million units are collected and transfused every year to tens of millions of recipients for whom blood transfusion is the only life-saving medical intervention. Interesting to say that the early days of blood transfusion, the very process of transfusing red cells was described as a transplant, which insists on the concept of red blood cell as an organ. As my name and my accent probably give away, I'm Italian. Unlike fine wine, unfortunately, red blood cells in the blood bag progressively lose the dispositive phenotype and start speculating and releasing vesicles, and the process ultimately results in the accumulation of morphological changes, leading to the formation of spherochenocyte and spherocyte, smaller than 42 square micrometers in projected surface area. Usually these spheres are much less deformable and are more likely to be sequestered in the spleen and ultimately untoward, promoting the untoward removal from the bloodstream of the recipient. How do we go about testing the quality of a blood unit to tell what percentage of these red blood cells is removed upon transfusion? One way that the Food and Drug Administration, the US and the European Council have agreed upon is to use radio-labeled tracers like chromium 51. The way this works has a lot of caveats and a lot of issues with the very test. First, for example, you're radio-labeling and then washing out the residual label, which in the process also removes the cells that are prone to lysis in the first place, which would be likely to be sequestered in the bloodstream of the recipient no matter what. And at the same time, you're also transfusing just 10 ml of blood in the whole unit in healthy autologous recipients. So the blood donors receives their own blood. And this test is not done routinely at scale for all the blood units issued in the United States or in Canada. It's only done for selected new blood products as they are trying to obtain CE mark or FDA licensing for issuance of new products. And to the standard criteria, with all the caveats that I mentioned, there's many more, but we don't have time to go much into details on that today, is that a good blood unit is any blood unit that for which the post-transfusion recovery, so at 24 hours, like the red cells that still circulate at 24 hours after transfusion, needs to be above 75%. So in other terms, you see that there's, across all, I think this was like over 20 studies that Dr. Dumono Bouchon have worked on for over a decade or two. Most of the donors actually meet the 75% threshold, which, by the way, 25% of the red cell, even a good unit, would have 75% of the red cells in a unit removed. A blood unit is approximately a tenth of the red cells in circulation, so we're looking at 2.5, 2.7, billion red cells per unit. So 25% is a quarter of that, which is now doubling, tripling, or quadrupling, depending on the math, the splenic sequestration capacity with a single blood unit not meeting the 75% threshold. More importantly, a series of studies on the age of blood said, well, yes, the age of blood impacts the post-transfusion recovery, but does it increase mortality? And a series of randomized prospective clinical trials suggested that at least there is no apparent benefit in transfusing the freshest available units as opposed to the standard of care. There are caveats and limitations to the studies. One noted that several of the studies were comparing day 14 versus day 21 blood unit when the shelf life is up to 42 days, so the spread wasn't that big. And ultimately, with the understanding that it really is a crude measurement to say, well, this blood unit is not as good because it doesn't kill you. There must be intermediate measurements and more refined measurements of whether a blood unit works properly or not. I never worked, I guess I postdoc for a while with the Italian National Blood Center, and my PhD was sponsored by the Italian National Blood Center. So I've been in blood banks for approximately 3 years and a half, but I never actually worked in a blood bank. So I'm looking at this problem more from the fascination of a basic scientist than an actual practitioner. So everything I'm going to tell you from now on is a pie in the sky of somebody who's done a lot of basic science, but perhaps is going to face reality that some of the things I'm going to be proposing are going to have to deal with the harsh logistics of keeping people alive. So blood saves people, but there is an heterogeneity in the distribution of post-transfusion performances, meaning how good the blood unit is across blood donors. Or in other terms, not all blood units are the same. Blood, like people, doesn't always age the same. Usually at this stage, I had a slide of Robbie Robson, who was a bodybuilder, age 79, and another person who wears a MAGA hat, age 79. I had to take those lights off not to be arrested by us on the way back. I'm joking, but not so much. This is not recorded, right? So how do we tackle this problem? Well, first we collaborate with a lot of people. I'm a firm believer in team science. I've been blessed with the people Jim is here in the room. We've been collaborating for years, who have been willing to involve me in their brilliant science and give me an opportunity to offer many nails for my hammer being mass spec-based metabolomics and proteomics. And in one such study, specifically the rats or recipient epidemiology and donor evaluation study, we've been collaborating with Dr. Mike Bush and the Vitalent Research Institute to test the biological and then eventually metabolic and molecular factors contributing to the heterogeneity of blood storage quality across 13,400, give or take, blood donors enrolled across 4 different blood centers in the United States. These donors donated a unit of blood, which was stored for up to 42 days. And at the end of storage, these units were tested for either the propensity of red blood cells to hemolyze spontaneously, which is another one of the quality control standards, gold standards for the quality of blood units, along with post-transfusion recovery. Pretty rude one, or more artificial tests like osmotic fragility or ping test, the standard ping test, to, for example, diagnose the hereditary spherocytosis, or the red cell capacity to withstand oxidant stress with AAPH pro-oxidants. In addition, these blood donors were also genotyped at 179,000 single nucleotide polymorphisms. And initially, the rats group and NIH had only funds to run 500 samples. And we ran that first, but then, in the meantime, we developed like ultra throughput ballistic methods to cut the cost exponentially and allow us to, without breaking the bank, perform metabolomics on all these samples, and ultimately use this to identify the molecular phenotypes of all these blood donors, which you ultimately can use this information to drive, to perform unsupervised statistical analysis and identify that there's literally a constellation, it kind of looks like a shooting star here, of different blood donors across metabolic states. What does any of this mean? Well, first, I didn't mention that for the donors ranking below the 5th percentile or above the 95th percentile, so the extreme in hemolysis parameters, good hemolyzer, bad hemolyzer, forgive me for the oversimplification, were invited to donate a second unit of blood. So this is the recall donor cohort of 643 blood donors. This second unit of blood was sampled again at day 10, 23, and 42, and tested again for all omics analysis of now 1,100 and some samples. There was a small cohort. And then of course, we have already the genotypes for them, and were tested again for hemolytic propensity. And once you do that, you see that there are metabolites that accumulate in the blood bag as storage progresses, and metabolites that are consumed in the blood bag as storage progresses. And ultimately, you can make a whole story identifying the so-called metabolic markers of the storage lesion, metabolites that consistently decrease or accumulate in a blood unit across all blood donors, independently of how well they store or how poorly they store, with the caveat that in some cases, some day 42 for some donors for some parameters rank higher than day 10 for other donors. Now we are finally, as we're starting looking at population scale level, understanding that yes, the metabolic storage lesion is follow similar trajectories across all donors, but the slope of those trajectories, the rapidity at which those lesions accumulate, changes on a donor-to-donor basis. And some donors at day 42 may look like donors day 10 and vice versa, which to some extent contributes to offering a rationale for explaining the lack of differences observed in the clinical trials on the age of blood. We can use this information to identify these metabolic markers of the storage lesion. Some of them have been patented. Jim developed something similar. Dr. Zimring here developed something similar for mouse studies. We and Dr. Bernard Paulson at the University of California, San Diego developed similar markers for humans, and we have now patents on this. And ultimately, just by measuring a subset of 8 metabolites, we can tell you with 99% specificity and sensitivity, whether somebody with our blood unit is a storage day 21 versus 42 and so on and so forth. And we can actually use the in silico modeling systems biology approaches that I was talking about at the beginning to predict omics trajectories, giving the snapshot at the time of donation for any blood donor, and even using all that information to come up with what could go bad in a donor as a function of storage duration, as a function of the proteome and metabolome at day donation. And ultimately, as I will show you in the next few slides, come up with ways to improve the quality of stored blood. To summarize this part of the presentation, during storage in the blood bank, red blood cells tend to deplete their energy reservoirs, including adenosine triphosphate and 2,3-bisphosphoglycerate. As ATP and DPG are fully consumed, the hemoglobin increases the oxygen saturation, hemoglobin, oxygen jumping on and off, more reactive oxygen species fentanyl hypervised chemistry, and ultimately accumulation of reactive oxygen species, lipid peroxidation, and oxygen stress. A process that we observe in these blood units. So, well, this is great, but what did we learn that we didn't know before, other than blood from some people stores faster than others, and the same things change, but change differentially. Well, we can use similar unsupervised approaches, but then perform a K-means cluster to identify donor endotypes based on metabolic signatures. And you see, for example, some donors that cluster very far away from any other group of donors. And if we focus on this cluster 4 random number that the algorithm pop out, we can see that actually these blood donors in this group have very high levels of adenosine triphosphate, and compared to the rest of the blood donor population, and donors clustering in this group have very low hemolysis compared to anybody else. Interestingly, donors in this group have higher levels of glycolysis throughout the lifespan. On the x-axis here, I'm showing the age of the donor now, not the age of the blood unit. And what we start observing is that based on donor biology, including the donor age or donor sex, biological sex, at least at birth, assigned at birth, we can see that there are differential metabolic trends for given metabolites. Here, for example, I'm focusing on glycolysis as the main and only energy generating pathway in mature blood cells. You can see, for example, for some metabolites like glucose, that we see declines across the age range for the blood donors from age 16 in this study all the way to over 85. We have sufficient sample sets, even though we have a donor at age 92, which is pretty incredible per se. I guess it's the healthy donor effect. I don't know if you'll have a chance to talk about that later. But the very interesting component here is that you see a chiasm between male and females, that the sex dimorphism is lost by age 51. We see that for so many metabolites. Age 51 is the average age for the menopause in the United States. So now we start observing that not only we have factors impacted by the storage age of the unit, but also by the donor age, the donor sex, and a combination with donor age. And ultimately, if you perform post-transfusion recovery studies, we find that the high energy phosphate compounds or their breakdown and domination products like apoxantin are positively and negatively associated with post-transfusion recovery as determined by chromium 51, or we can actually link the metabolite level to hemoglobin increment and bilirubin increments in transfusion recipients. Through collaboration with Dr. Narek Rubini at Kaiser Permanente, we could determine the hemoglobin boost, the delta, adjusted delta, because there are caveats there, how much hemoglobin levels increase after transfusion in recipient of blood units with the high levels derived from donors with high levels of ATP versus, for example, high levels of hypoxantin. So now that we are appreciating that, for example, there are factors like biology, such as, for example, donor age, BMI, sex dimorphism, the other solution that was used to sort the blood cells, blood centers, we now start observing that there are so many factors that contribute to the heterogeneity of blood units as a function of storage. How do we control for those factors? One way to go about that is to use, for example, model organisms. One in particular developed by Dr. Zimring allows to test any blood cell of interest by transfusing into mice expressing UBGFP in all the red blood cells, and then using fresh M-cherry, so red labeled red blood cells in as a tracer control. And ultimately, we can use that to test, for example, the post-transfusion recovery in mice across different mouse strains. Now you can adjust for mouse age, mouse sex, diet, and other exposures, and ultimately just focus on the impact of genetics, and ultimately identify some mouse strains that store really well, some mouse strains that store poorly, and you can cross them for multiple generation. For example, here, by looking at the Jackson Lab diversity of bred population of 8 founder strains, including some domesticated ones and some wild strains, and obtain up to, in the first study, 350 mice, and the second study up to 525. And then what you can do is not only measure the post-transfusion recovery, but also the proteome, metabolome, lipidome, and link it to post-transfusion recovery, and link it to the genotype of these mice, as they are also genotyped at approximately 140,000 polymorphisms. And ultimately, you can build a whole infrastructure of gene, metabolome, proteome outcome, like post-transfusion recovery association. And if you're interested in more about this, actually the papers are published, and the QTL viewer is available for public access through links, or you can just take a photo of the QR codes. For the sake of this presentation, what we did find is that the center of this network, gene, metabolome, proteome, and outcome network, we found in particular a ferry reductase TYP3 that is co-mapping with a series of metabolites, lipids, and lipid peroxidation product oxylipins on chromosome 1. And the higher lipid peroxidation, the lower the post-transfusion recovery, and the different SNPs variants of this ferry reductase TYP3 that converts ferric iron to the ferrostate, in so doing promotes a right shift of phantom and Haber-Weiss chemistry towards the formation of reactive oxygen species. You can cross the mice, you can isolate. I think it took like 7, 8 generations maybe to isolate a 4 kilo base region on chromosome 1. So these mice here are fully black 6, except for a 4 kilo base region on chromosome 1, and ultimately validate that SNP3 is associated with this lipid peroxidation, low post-transfusion recovery phenotype. Does any of this matter in humans? We can go back to our rats cohort and see that we actually have multiple either missense or regulatory polymorphism of the region coding for SNP3 that are linked to the different degrees of hemolysis. And this is emerging from a target analysis if you want to hypothesis test in humans, but also if you do a completely untargeted, unsupervised analysis linking hemolysis to lipid peroxidation product in humans, you may not find necessarily SNP3, but you find a series of other polymorphisms in mostly regulatory polymorphisms in all the enzymes involved, not necessarily in iron reduction, but in the reduction of lipid peroxidation products and recycling lipid peroxidation product through processes that, if you look closely, and any of you works on ferroptosis, closely recapitulate ferroptosis or death by iron-dependent lipid peroxidation. We can actually link all of this again to hemoglobin increments. The higher the lipid peroxidation, or the higher the SNP3 or the polymorphism on rate-limiting enzymes on the damage repair pathway like LPCAT3, the lower the hemoglobin increments in recipients of blood units from these donors. One thing that we can do then is we can get red blood cells from, we can look at the endostorage red blood cells in a blood unit. You literally have a mixed bag of red blood cells. Some of them are morphologically altered, some of them are not. What you can do is that you can actually incubate with the fluorescent substrate, and the red blood cells that are metabolically active and morphologically stable actually manage to pump out or do not incorporate the fluorescent probe, while the others are keeping it. And you can actually positively enrich through flow sorting the poor morphology red blood cells with the good morphology red blood cells, and then perform metabolomics and proteomics and lipidomics analysis. And once again, the red blood cells that are altered morphologically, metabolically, are the ones that accumulate the most lesion, most energy depletion, most lipid peroxidation, and so on and so forth. To the point that if you look specifically at the population, they look like they completely depleted the carnitine pools, which is essential for reacylation of the lipid peroxidation product upon formation of oxylipin and detoxification into lipid alcohols through the so-called Lent cycle. So if that is true, what can we do about this? Well, first, test whether any of this is relevant. We have these cells that are differentially labeled in a mixed bag of different population, and we can show that the ones that are morphologically altered and fluorescent labeled are the ones that are preferentially sequestered in ex vivo perfusion models of human spleens from organ donors that are ex vivo perfused with fresh and stored red blood cells. Incidentally, all of this matters to red blood cell biology beyond storage biology, because as red blood cells age in circulation, they similarly deplete acylcarnitine pools, and the capacity to reacylate oxidized lipid peroxidation products and lysophospholipid derived from the phospholipase A2 dependent activity on these oxidized lipids is lost as the red blood cells age in circulation, as determined by biosimulation studies and Perkel density gradients in humans. We can use all this information in metabolomics, proteomics, lipidomics to map in silico a model of red blood cell membrane morphology, and ultimately predict that the red blood cell at the end of storage have increased bending, altered deformability and increased bending rigidity. Pretty much are less deformable, are more likely to be sequestered in the spleen, as our experimental data shows. So what do we do with all this information? Well, one thing we can do is, for example, replenish the L-carnitine pools. We can also use the non-active stereoisomer D-carnitine, and it does nothing, while the L-carnitine rescues the phenotype, boosts L-carnitine pools, and almost doubles post-transfusion recovery. We can target a key rate-limiting enzyme of glutathione-dependent detoxification of lipid peroxidation product, like GPX4, and ultimately, we can rescue the phenotype of lipid peroxidation and post-transfusion recovery with inhibitors of ferroptosis. Specifically the one that target the mature protein rather than the expression and stabilization of the GPX4 enzymes. We can use antioxidant, lipophilic antioxidants like vitamin E in combination with L-carnitines to establish the phenotype. So I still have a lot of slides, and I don't think I'm going to go through all of them, but for the sake of today's presentation, I'd like to show you a few more things. As we're studying like all these metabolites that change with storage, I probably convince you that depletion of energy pooling, accumulation of oxygen stress are both bad signatures for red blood cells. But there are also factors that are completely independent of storage duration. For example, small molecule metabolites like canurinin that do not change at all in as a function of storage duration from day 10 to day 42 in the red record population, but they tend to be the top predictor of osmotic fragility as gleaned by, for example, random forest and elastic net modeling. And this metabolite is not really synthesized in mature blood cells that lack the enzymatic machinery to make them, but it's being uptaken by red blood cells from the bloodstream and recapitulates what happens in the bloodstream of the donor at the time of donation. And some of these metabolites, like for example, canurinin, are proportionally increasing with the donor age as a function of the BMI. So the older, the higher, and the heavier the BMI of the donor, the higher the levels of canurinin. We have sex dimorphism again through the age of 51, the average age for menopause. And ultimately, you can build like all sorts of age-related signatures of metabolites that accumulate or are depleted with age. And of course, I don't have the time to go through that today, but we have published a few papers on that on Aging Cell and other similar journals. The really interesting aspect of this metabolite is that even though they do not change with storage, their levels are reproducible across multiple donations from the same donor. This is, for example, the canurinin level in 643 index donors who are also part of the recalled population, suggesting that there are donor-specific signatures associated with these metabolic phenotypes. or ultimately donor genotypes linked to these phenotypes. In particular, for example, for kynurenine, we see that the transporter or rate-limiting enzymes for the synthesis or degradation of kynurenine are all mapping with the kynurenine levels across 13,000 blood donors. And ultimately, we can use this information to build gene metabolite association network in humans to establish a novel map of the red blood cell genome metabolome and literally rewrite the textbooks for red blood cell metabolism. At this stage, I'd rather thank you all for your attention and leave the floor open for any question. And I apologize for having included way too many slides, but there was 50 out of 170, so we have plenty of things to discuss if you want. No, I mean, the actual presentation was just another 5 slides long, but thank you so much for your attention.
Yes, so trophic biochemistry a bit beyond me, but at a practical level, you're identifying people whose red cells have, in theory, a superior red cell survival. Now, one group that's been a real problem over the years are people who require multiple persistent transfusions, and that would be the thalassemics. Are you at a stage where you can identify a donor to match, if you like, a vulnerable recipient because of iron toxicity?
You know, it's part of the presentation that I didn't have a chance to talk about, but we know, for example, that some categories of donors, especially for, you know, very similar genetic ancestry, overlap with the recipient population that they're serving with their blood to donate, tend to be matched at a non-random, non-stochastic fashion. So now we have, for example, traits that are linked to increased hemolysis like GXPD deficiency, right? That are more preferentially transfused to recipient with the sickle cell disease, for example, just because of the African ancestry, for example, being prevalent in donors of African descent who carry the African variant and recipient of that blood. And we actually did like preliminary clinical trials and post-transfusion recovery studies with Chromium 51 in patients with sickle cell disease, and show that even just receiving a transfusion of a single unit of GXPD deficient trait is associated with an 8 to 15% lower performance in terms of post-transfusion circulatory capacity. And we argue in terms of hemoglobin increment, even a 20-30% loss in efficacy. And in mouse models, we can actually look at tissue oxygenation. We can bleed the mice and transfuse them as you go, like we do, for example, in our trauma patients. And look at what happens to tissue oxygenation. And we have, for example, increased tissue hypoxia in the subjects, in these mice models. The point you were making is that, if I got it correctly, is that you are now matching the thalassemia, for example, recipient with the red cells with a higher hemolytic propensity, and you're making them more susceptible to untoward complications, right?
Yeah, the big issue is to get the maximum red cell survival to minimize the iron toxicity. And certainly you've given us some biochemical markers. There's a question of, is there anything fairly simple to screen donors? Because this is beyond the average sort of screening process.
Well, first of all, we believe that we can run our metabolomics at scale at a cost that is sufficiently low that we could theoretically do that for all blood units that are issued in Canada every year. I really do believe that. That's one of the reasons why I'm here. So once you have that information, of course, like now we got to the point where we accumulated enough basic science knowledge that you know, we either test the hypothesis or be done with it, right? And I think I cannot see any better system than a universal socialized healthcare system that both has the health records for the donors and the health records for the recipients that allows such vision to be implemented in the real world. At the net of that information, I mean, there are factors like that are emerging from these studies, including, for example, these otherwise healthy blood donors. When we start doing like gene metabolome association in the blood donors, this is from rats. And we link, for example, metabolite in gray to gene with specific polymorphism as they map through MQTL analysis. We see that for several metabolites across multiple pathways, the gene for which we find relevant polymorphisms are established target of known, relatively frequent polymorphism links to inborn errors of metabolism. I mentioned G6PD deficiency. We see healthy blood donors who carry sickle cell trait. We see healthy blood donors who carry pyruvate kinase deficiency trait. Of course, they don't have overt manifest disease, but the stress of blood storage is sufficient to unmask the metabolic phenotype and the gene metabolome interaction. And what we believe is that, of course, this is irrelevant, whatever we find for population that carry those traits. We've done some studies on pyruvate kinase deficiency, hypoxia disease, /NIAN, you name it, all sorts of important error metabolism. But also understanding how to improve the blood unit may potentially theoretically contribute to improve the health of the blood donor over time, as for example, aging or epiphenomena, exposure, getting in a car accident and almost bleeding to death would unmask the signature, the phenotype that we had already observed by looking at how their blood ages in a blood bank. So in other words, not only would we do a benefit and a service to the recipient by looking at the blood unit, but we believe also to the donor by looking at their blood and linking the blood metabolome to genome signature in a way that would be telling of how their blood would respond to perturbation, of which blood storage is just an in vitro model. It's an oxygen stress model that would unmask other phenotypes. One of the phenotypes we believe we unmask is the age-related phenotype. Like, for example, we can use the metabolome proteome signatures to build literally aging clocks, where we can feed the metabolome, the metabolites that change with age and the protein that change with age to machine learning algorithm, random forest, elastic net, you name it, and ultimately be able to predict just based on the metabolic signature or protein signatures somebody's age. And then we can tell the deviation of their predicted age from the molecular phenotypes from the aging clock. So whether the clock is ticking faster than their actual age, whether they're aging faster than they actually should. And we see that, for example, people with susceptibility to hemolysis, potentially poor storage, are more likely to age faster. And not only that, I think this hopefully potentially mic drop. We can use, again, this will have to be validated prospectively, right? But we now have signatures that predict with 70% specificity and sensitivity whether a donor is going to be donating blood 12 years from now. Because, rats, the samples were collected 12 years ago, and at Vitalant, Pittsburgh and San Francisco, we still have records of who's still donating blood, who got deferred for all sorts of purposes. I'm still getting access to the IRB to understand what were the purposes. But we can, over the time, we have the longitudinal numbers of donation. We now have pretty clear signatures that can tell you whether a donor is going to stop donating blood in the next 12 to 24 months fairly quickly. Whether other donors, stop donating blood, but they have signatures that are consistent with being able to still be donating blood. So maybe you can have targeted donor recruitment or retention campaigns that specifically target these donors rather than these donors. And ultimately, give something back to the donors. Like we believe that now we have signatures of what, I'm borrowing language from Eric Topol. I understand it's rather sloppy, but if you want to call somebody a super ager versus a poor ager, somebody whose molecular age is delta 15 versus a delta negative 15. So they look 10 years, 15 years older than what they actually are, or 15 years younger than what they actually are. They are twice more likely to be donating blood. So we want to keep our donors younger, maybe, so that they keep donating blood, saving lives, at the same time, maybe improving their own lives. I'll spare you all the continuum of health concept from blood donors to athletes to patients with all sorts of diseases, but the concept is almost self-explanatory. Collaborating with any and all of you, there may be a continuum of health across diseases that the molecular signature, molecular fingerprints at the metabolic level, for example, can inform whether a signature we see in a blood donor could be predictive of an early onset of a given disease. We're talking before about neurodegenerative diseases. What could be transfusion transmittable, other transmissible, or whether, for example, intervention in lifestyle, in exposure, diet, drugs, supplements, or even just exercise can keep you young. And we have, I think, the tools at the molecular level to monitor all of this. Again, at this stage, it's correlative, but will have to be tested prospectively.
Okay. Hey, yeah. Thanks so much for that wonderful presentation. It's always great to see always great to see your work. And so I'm interested in this idea of populational level metabolomics, because I mean, you can get a lot of information about a person's lifestyle, and it's almost like like their Google search habits. What do you foresee some pushback from sort of the privacy side of things about taking that much information from so many people?
There may be different layers of approaching this problem, and we acquire data in an untargeted and supervised fashion, so all the data is there, but we can look only for some things, right? So we can come up with a targeted metabolic panel that ignores all the other metabolites that are there and specifically pick and report the peaks of interest, dismissing all the other information in a way that does not cross boundaries related to ethical approval. That said, yes, small molecule metabolite exposures are there. Believe it or not, like we see a lot of caffeine in a blood unit, but then, we can tell whether somebody smokes versus they, in Colorado, it's hard to find a blood unit that doesn't have THC and CBD, right? Or, resveratrol, thinking of red wine lovers versus, other metabolites that derive from the diet, whether they are vegan, methylhistidine versus other, metabolites of plant origin. What we found really fascinating, and we proposed the concept of the red blood cell blood donor exposome, is that, whether we report these metabolites or not, they're there. And, for example, we see in blood units drugs that are not ground for blood donor deferral. We see, for example, Zartans, blood pressure medication. We see statins. We see acetaminophen, NSAIDs, ibuprofen. We see sorts of things all the way to, I'm going to make you smile. I told you that we had several 80-year-old or older blood donors. Well, in 3 85, 82, and 87-year-old male donors, we found sildenafil, Viagra. And, the blood looked amazing. Not telling that, not saying that everybody should get Viagra before donating blood, but there are, whether we like it or not, these molecules are modifiers of blood. And of course, we don't know whether it's just an association between the quality of blood and the presence of molecule compounds. We don't know whether the abundance is sufficient to be of relevance to the transfusion recipients in terms of safety. There are some reports, we were talking about this over lunchtime, of blood transfusion recipients receiving blood from donors who ate peanut, and the recipient suffered from peanut allergies and went into anaphylactic shock. There's I think 1 or 2 reported cases in the literature. What I can tell you, for example, for some of the easy targets like caffeine, the most abundant metabolite of exposure we see across almost all of donors, Historically, we'd been telling donors drink coffee because it helps with vasovagal syndrome. There's some iffy literature there, and it boosts blood pressure. Probably not a problem in Canada, but in the United States, the blood centers are non-profit, but they are paying for the people, the clinician where they're collecting blood, and they want to collect as many units in the shortest amount of time possible to cut cost. And they recommend drinking coffee before going and donating blood. The hypothesis, the idea is it's going to help you with the vasovagal syndrome. That practically just boosts by your blood pressure faster. But then we see that like if you drink coffee, you have a decent amount of reproducibility of caffeine level across multiple donation. Part of it is explained by NQTL mapping on cytochrome P450 reductases that metabolize coffee itself into xanthan, methylxanthan. But then there are polymorphism. The caffeine also act as an agonist to the very adenosine receptor I mentioned when I was talking about the high altitude studies, because it's a purine analog. Now, by the same token, stimulating adenosine receptor boosts your capacity to make 2,3-DPG. Caffeine inhibits it by inhibiting adenosine receptor stimulation and boosting activation of the pentosphosphate pathway. However, caffeine can also penetrate red cells through ENT, the same transporter for adenosine, and it's a direct inhibitor of G6PD. And now, red blood cell from somebody who drank coffee within 6 hours before donation look like a G6PD deficient red blood cells. So that's one example of exposome, metabolome, genome interaction that could be a potential translational relevance. And we tested this mechanistically in the mouse model, and some of us drinking coffee and donating blood and testing it in vitro.